In this video, I'm going to walk you through this diagram. To begin, let me orient you to the muscle cell at rest. Note that this brown line at the top represents the sarcolemma of the myocyte, or the myofiber, or the muscle cell. Note that at rest, the cell is going to be not actually at rest. We have these sodium-potassium pumps that are constantly pumping 3 sodiums out for every 2 potassiums in, and of course some of that potassium is allowed to leak back out, leaving a lot of negative stuff left behind in the cell. Collectively, the actions of all of these different pumps and channels is going to generate a resting membrane potential of negative 85 millivolts, so very negative, and so that is existing throughout this entire cell. Okay. Also note that the sarcolemma does not just remain on the surface of the cell. Instead, it has these deep tubular invaginations through the middle of the muscle cell. These are called T-tubules, and they are flanked on either side by the sarcoplasmic reticulum. Specifically, the portion of the SR that is closest to the T-tubule is called the terminal cisterna for singular, or terminal cisternae for plural. Terminal cisterna plus T-tubule plus terminal cisterna is collectively called the triad. Now, also at rest, the muscle cell is actively pumping calcium out of the sarcoplasm and into the SR, so that there is ultimately a very steep concentration gradient across the SR membrane, so in this sarcoplasmic reticulum versus within the sarcoplasm. Also, I want to point out that the T-tubule is lined with voltage-gated sodium channels, just like the surface of the cell, and that there are voltage-gated calcium channels that are connected to both the T-tubule membrane and the sarcoplasmic reticulum membrane. Okay. Other features of the sarcoplasm. Remember that most of the sarcoplasm is filled with cylinders. These cylinders are called myofibrils, and within them are overlapping myofilaments. The bluish purple here is actin. Remember that actin proteins are connected to one another by Z discs, and so from Z disc to Z disc, this is called a sarcomere, and it is the contractile unit of the muscle cell. Overlapping with the actin, we have myosin. Remember that myosin has these heads, okay? And at rest, these myosin heads are prevented from attaching to the actin's active sites by another type of protein, seen here as this squiggly teal line. The protein blocking those active sites are tropomyosin molecules. Okay, so again, at rest, when your muscle is not contracting, tropomyosin is going to prevent the interaction between myosin and actin. The only way to remove tropomyosin is by binding calcium to these little pink molecules here, which are called troponin. Troponin regulates whether or not tropomyosin is blocking the active sites and therefore allowing or preventing what are called cross bridges between the myosin and the actin. So now let us stimulate the muscle cell. Okay. Here is a motor neuron, which we've talked about before. Step 1 is an action potential traveling down the motor neuron. As we know, calcium floods into the cell at the synaptic terminal and leads to the exocytosis of acetylcholine from the synaptic vesicles of the motor neuron. The acetylcholine then is exocytosed into the synaptic cleft and is going to bind to the receptors located on the motor end plate, the squiggly line populated by acetylcholine receptors. Now, step 2 is when this interaction between acetylcholine and its receptors allows the influx of sodium into the local area. This is going to take the very negative resting membrane potential and start to depolarize it. If there is enough of a stimulus to reach threshold, this depolarization is going to trigger step number 3, which is the opening of these voltage-gated sodium channels, allowing a further influx of sodium into the cell, therefore depolarizing this area of the cell and stimulating an action potential. So this yellow squiggly line here is an action potential traveling actually out in all directions from the neuromuscular junction. Okay, so I'm only showing it over here, traveling along the surface of the cell, but keep in mind that this action potential is also going in this direction and in all directions around the muscle cell. Specifically, this action potential is also going to travel down the T-tubules. Remember that the T-tubules are essentially the sarcolemma, but traveling through the inside of the muscle cell. Therefore, this membrane potential, this difference in charge across the membrane, is going to be the same in this T-tubule as we have the difference up here on the surface of the cell. Now this depolarization, this action potential, is going to lead to step number 4. Step number 4 is the opening of these voltage-gated calcium channels. All right, so we hit the right voltage to actually open up these channels, which I'm trying to show here. All right, so this channel is now open, and calcium is going to diffuse out of this channel down its very steep concentration gradient, so this is a very fast process, and it is essentially going to be released into the sarcoplasm. Okay, so keep in mind that the SR is actually covering over all of this stuff over here. I haven't drawn it just because it would be very confusing if I did, but keep in mind that as calcium exits out the SR here, it's actually bathing over the myofibrils and the myofilaments within. Okay, so as we know, calcium, when it binds to troponin, is going to stimulate troponin to move, to physically roll tropomyosin out of the way, which is what we can see here. Here, tropomyosin is covering the active sites, and so the myosin heads are unable to interact with actin. But as soon as calcium binds to troponin, troponin rolls tropomyosin out of the way and allows myosin heads to bind to the active sites on actin. This is the formation of a cross bridge. So it is a bridge using the myosin heads between this red myosin and this bluish purple actin. Okay, so we can see a couple cross bridges here because calcium has caused troponin to remove tropomyosin from the active sites. Now, if we zoom in right here, we can explore step number 6. Step number 5 is actually triggering step number 6A, which is the beginning of the cross bridge cycle. So I have drawn the cross bridge cycle for you down here. Remember that we are talking about this area. Okay, so as long as tropomyosin is out of the way, this myosin head is going to bind to the active site, or the active sites on actin. Now, just for a reference here, I have drawn in a couple stars. Note that this star, so this pivot point on the myosin molecule, is off to the left of this particular point here. Now, as soon as this cross bridge forms, what's going to happen next is called 6B, the power stroke. Okay, so essentially this entire molecule pivots and essentially pulls the actin in this direction. So now this point, right, this point up here is pretty much right on top of this point over here. So we have actually moved this entire structure, this entire molecule towards the M line, towards the middle of the myosin. Okay, so that's the power stroke. In order to actually fuel this conformational change, we actually get rid of ADP and phosphate. Right up here, we can see ADP and phosphate are attached. We'll get to that in a second. The power stroke is fueled by essentially getting rid of these two molecules. That changes the shape of myosin and pulls the actin towards the M line. Next, the cross bridge has to break. Okay, so in order to do so, we use ATP, right, this energy-rich molecule, to break the cross bridge, which used to be right here. Okay, the actin and myosin are still in the same places relative to one another, and it's important to note that the ATP is the energy, so the actual active part of this process happens to be breaking the cross bridge. Okay, the ATP is very soon broken, right? So we hydrolyze ATP into ADP and inorganic phosphate. And essentially what this does is it changes the shape of the myosin head once again into its cocked position. So now it is ready to bind to the next active site on actin as long as tropomyosin hasn't covered over it. All right, so as long as the muscle is still being stimulated, as long as there is still calcium out in the sarcoplasm, this myosin head is now going to bind to the next active site. Right? With its ADP and phosphate, we have formed a cross bridge. As soon as the cross bridge is formed, we get another power stroke. ADP and phosphate leave, opening up a spot for a new ATP. ATP breaks the cross bridge. When ATP is hydrolyzed into ADP and phosphate, the head is recocked and the cycle begins all over again. And so this is going to continue until the cell is repolarized, ACH is removed from the synaptic cleft, calcium is pumped back into the sarcoplasmic reticulum, troponin allows tropomyosin to roll back over those active sites, and therefore this recocked myosin head is unable to form a new cross bridge. And so then the sarcomeres would slide back to their original configuration because of the titin molecules at the end. And so that is an overview of this diagram and the process of muscle stimulation and contraction.